Good morning, children. The experiments performed by Griffith, the transforming principle for the search of the genetic material, and later on for this topic only for the search of the genetic material, the experiments were performed by McCarthy, McLeod, and Avery. Still, not all biologists were convinced for the fact that DNA is the genetic material, so they were unequivocal for this that genetic material is DNA. So later on, Hershey and Chase he carried on with the experiment in 1952, and he performed his experiments by taking bacteriophage. What are bacteriophage? These are the viruses which attack bacteria. So this is the structure of a bacteriophage, roughly drawn. The outer portion is known as capsid, which is made up of protein, and inside is the genetic material. So one group he grew the bacteriophage in sulfur radioactive, and one group he grew on phosphorus radioactive. Sulfur is the content of protein, that means this part of the bacteriophage will become radioactive, and phosphorus is the part of DNA, so genetic material will become radioactive. This he did this experiment to find out that which part will become radioactive. Now we know that here the protein coat capsid will become radioactive as sulfur is the part of protein. Now he grew this on bacteria E. coli. So this is bacteria E. coli, and bacteriophages will attack this and genetic material will be introduced. It is quite obvious that here the genetic material was not radioactive as it was grew in radioactive sulfur. And this side the genetic material, as the bacteriophage attacks the E. coli, the genetic material is introduced inside, and as this bacteriophage was grown on radioactive phosphorus, so this genetic material will carry radioactivity characteristics. So now what? The second process is to separate this bacteriophage from the bacteria. This phase is known as blending. This is done to separate the bacteriophage and the bacteria, which is the second step. Now these two are separated. Now the third step is centrifugation. Now what is done in this? Why is this done? Because of the high speed, the cells will separate from the supernatant liquid. Once the cell will separate from the supernatant liquid, here he found that the cells were not radioactive. Why? The bacteria cell did not get any of the radioactive because genetic material was introduced by this bacteriophage which was not carrying any radioactivity. But the supernatant liquid, it became radioactive because there the protein coat was in the supernatant liquid. This side what happened? This side, as the genetic material was grown in radioactive phosphorus, here the cells were radioactive and the supernatant liquid was not radioactive. Hence this experiment proved that the genetic material is DNA. Final proof was given by Hershey and Chase. Thank you, children.